Welcome back, everyone. So now you can tell me how a bacteria can get into your patient's body, how it can avoid the immune system, how it can stick to their cells, how it can get inside of their cells. You can describe to me how it could cause disease, how it could take up iron, how it could produce exotoxins or endotoxins, and you can compare those two different types of toxins. But of course, bacteria aren't the only thing that can cause disease. So let's start exploring some other types of microbes. First, we're going to talk about viruses. If you remember from our previous discussions, viruses are very different from other organisms. They are very tiny because they're not made of cells, and they're too small to be able to see with a microscope. So if you can't see them, you're going to be looking for other signs of an infection. And these are called cytopathic effects. Cytopathic effects are visible effects of a viral infection on a cell. So the virus is doing something to the cell, and the cell is going to look different. Because of the viral infection, the cell looks different. And so you can look for those cytopathic effects to identify a viral infection even when you can't find the virus itself. One example of a cytopathic effect is stopping cell synthesis. So cells are not being created when they normally would be. One is by causing lysosomes to release their enzymes and wreak havoc in that cell, break down. Who knows what could be broken down in that cell? One is by losing contact inhibition in a cell. Contact inhibition is the ability of cells to recognize that when there's a wound, they're not touching other cells, so they need to grow to fill in that wound. But when they have grown to the point where they are touching each other, where they have contact with each other, they stop growing because there is not a wound that they need to fill in. So contact inhibition makes sure that cells do not grow unless they need to be. If a virus makes a cell lose contact inhibition, that cell is going to start growing inappropriately. It is going to grow when it shouldn't be. That could lead to something minor like a wart. It could also lead to something very much more involved, such as cancer. Some viruses can create inclusion bodies. That's what we're seeing, these spots in this cell. These inclusion bodies might just be clumps of viral particles, just kind of clumping together as the viruses are forming. Sometimes viruses, this one's really weird, I think. They can cause cells to fuse together. So instead of having a small cell with a nucleus and another cell with a nucleus, I'm sorry if you can hear my cat playing the piano in the background. So instead of having these individual cells with an individual nucleus in each of them, these cells are fusing together to make this huge cell with a lot of nuclei inside of it. So it's multinucleated. It has a lot of nuclei from each of those cells that were fused together, and it's huge because it's a bunch of cells together. They might induce antigenic changes on the cell surface. That means that that cell is producing different antigens. It won't react or bind quite the same way as it normally would. And it might change the host cell function or change the chromosomes. Some viruses are able to stick themselves right in the middle of DNA, and so you would have a slightly larger piece of DNA. Or it can turn on genes or turn off genes that should not be turned on or off. And so you have chromosomes being activated or inactivated inappropriately. One very important thing that viruses can do is they trigger the formation of interferons. So alpha and beta interferons are chemicals that are produced by a virally infected cell. So once that cell is infected with the virus, it's toast, and it knows it's toast. So it doesn't really try to save itself at that point. It tries to save the cells around it. So it sends out a message that says, hey, I've been infected. If you've not been infected yet, there's still hope for you. Protect yourself. So it sends out this interferon message, these alpha and beta interferons. They go to the neighboring cells, so those neighboring cells can protect themselves from a viral infection, so that you have some cells remaining to be able to survive the infection. When that interferon is received by the neighboring cells, they will start inhibiting, they'll be able to inhibit the synthesis of viral proteins. And it also triggers for virally infected cells to be killed by apoptosis. So that cell that is sending the interferons is also kind of signing its own death warrant because it knows it's going to be killed by apoptosis, which means that it is going to be nicely broken down instead of a big old necrotic explosion of, a mess of viruses going everywhere. So now you can define cytopathic effects and give examples of it. And let's talk about fungi. So sometimes a fungal infection causes symptoms just because of the waste products of the fungus growing there. Sometimes if there is a chronic infection, if you remember, chronic infection means a long infection, it can produce an allergic response. Some fungi, such as Fusarium, can produce trichothecene toxins that will inhibit protein synthesis, so your cell is not making the proteins it should. Candida and trichophyton can create proteases that can affect your host cell membranes, and Cryptococcus can create a capsule that prevents phagocytosis. So some very similar themes to what we saw talking about bacteria, but in its own kind of own fungal type of way. Claviceps purpurea is a fungus that grows on grains. It produces a toxin called ergot. Ergot is a toxin that is a natural source of LSD. So it has symptoms very similar to someone who is doing drugs. It can cause intense hallucinations, delirium, and psychosis. This is what caused ergotism in Europe in the Middle Ages. This is a fungus that could grow on those rye and cereals, and people would eat it and then have these hallucinations. There is some evidence that it might be related to the Salem witch trials, that, people were having these hallucinations that led them to actually believe my neighbor actually is a witch, or my neighbor actually did, turn into a frog or whatever. Today, modern milling practices are able to prevent the growth of this, so you should not have a problem with this. However, if you do have moldy cereal, throw it out. Don't eat it. You don't want to, hopefully, you don't want to have this in your body. Aspergillus flavus is a mold that sometimes can be found in peanut butter. So if you have moldy peanut butter, also throw it out. Aspergillus produces a toxin called aflatoxin. I also always feel like I'm like a duck in the Aflac commercial. So it produces aflatoxin, which is carcinogenic. That means that it can be altered in your body to start producing mutagens. So it can mutate DNA and cause cancer. And Amanita phalloides, technically not a microbe, I know. But Amanita phalloides is this death cap mushroom that produces phalloidin and amantin. And these are two neurotoxins that if they are ingested, a neurotoxin tells you it's going to damage your nervous system and will lead to death. We will pick up with the rest of this chapter in our last lecture video. So I will see you in a little bit. We'll talk about protozoans, amoeba, helminths, and then what's happening, how are they going to spread out of your body. See you later. Bye.